My name is Louise and I'm here today to talk to you about Equine Coat Colour Genetics. I'm currently studying equine science at Charles Sturt University and hoping to work with horses when I graduate. The first thing we need to know when talking about genetics is what is genetics. Genetics are the blueprints for living things. They determine what you look like, how you grow, and how your body works. They are inherited, which means that in animals, genes are passed down from parent to offspring. Approximately half of the genetic material comes from each parent, though each individual gene has equal chance of being passed down from each parent. Some genes are located close together on the genetic material and are more likely to be inherited together. They are called linked genes. Some traits are controlled by multiple genes that can be inherited separately. Other traits may be influenced by the environment as well as by genetics. New genes are formed by mutation of existing genes. In wild animals, new genes can increase or decrease the likelihood of an animal passing its genes on to the next generation. This is known as natural selection. When people choose to breed animals, they can artificially select for or against specific traits. This includes colour, in order to produce a desired offspring. When living things' genetic code is known, it's known as a genotype, and the way it presents is the living thing's phenotype. When we talk about genetics and genes, we use specific annotation to denote what the genetic code is. The first is when a gene has two forms, a dominant and a recessive form. An uppercase letter is used to denote the dominant gene and a lowercase letter is used to denote the recessive gene. Secondly, when a gene is either present or absent, we use the gene code if it is present or the letter N if it is absent. If the gene is not known or does not influence the phenotype, we use an underscore. In horses, coat colour is first determined by the base colour, then dilutions, then white patterns, and finally by grey status. Some colours and patterns are breed specific and others may be found in many breeds. The base colour of a horse is made up of two genes, extension and agouti. Extension uses the letter E to denote the two versions of the gene. If the horse has at least one dominant extension, the horse will produce black pigment. If the horse has two recessive extensions, they'll only produce red pigment. Agouti is a simple dominant gene and is the restriction of where that black pigment is expressed. Agouti uses the letter A to denote the two versions of the gene. If the horse has at least one dominant agouti, the horse will have restricted black pigment, so it'll only be at the points, the mane, and the tail. And a horse has double recessive agouti, they will have unrestricted black pigment, meaning the entire horse will be black. These two genes combine to make up the three base colours of the horse. Chestnut horses have two recessive extensions, meaning they can't produce any black pigment, so the agouti status doesn't matter. Bay horses have at least one dominant extension and at least one dominant agouti, and black horses have at least one dominant extension and two recessive agouti. Chestnut horse has only red pigment and is genetically lowercase e, and underscore for the A. Some breeds may have different names for a chestnut, such as red or sorrel, and there are shade variations such as liver chestnut and flaxen chestnut, however they are all genetically chestnut. A bay horse has both red and black pigment and is genetically at least one dominant extension and one dominant agouti. There are shade variations of bay, such as blood and dark bay, however they are all genetically bay. A black horse has only black pigment and is genetically at least one dominant extension and recessive agouti. Horses that are described as brown might actually be really dark chestnuts or bays, or sometimes they could be sun-faded blacks. The first of our dilution genes is cream. Cream dilutes only red pigment, whereas 2 copies of cream dilutes both red and black pigment. So a chestnut horse with 1 copy of cream is a palomino. They have a lighter mane and tail as well as the body. A bay horse with 1 copy of cream is a buckskin. They have a lighter coat in their body but maintain the black points, mane and tail. A black horse with 1 copy of cream is a smoky black and they still appear completely black. 2 copies of cream results in double dilute where an entire horse is a pale goldish colour. A chestnut with 2 copies of cream is a perlino, a bay with 2 copies of cream is a cremello, and a black with 2 copies of cream is a smoky cream. Cream is found in a lot of different breeds, including Spanish breeds such as the Andalusian and Lusitanos, as well as quarter horses and paints, which do have Spanish ancestry. Pearl is the next dilution gene. It's on the same location as cream, so a horse can't have 2 copies of pearl and 2 copies of cream. It can only have a total of 2 of both genes combined. So pearl by itself has no effect, but either pearl with cream or double pearl can produce similar effects to double cream. So chestnut with 2 copies of pearl is known as a chestnut with double pearl, bay is bay with double pearl, and black is black with double pearl. Pearl is found in breeds with Spanish origin again, such as the Lusitano and the Andalusian, as well as derived breeds such as paint horse and quarter horses. The next dilution is dun. The dun gene has 3 variations: dun, not dun 1, and not dun 2. Dun causes dilution of the coat and primitive markings, dorsal stripe, legs, and ear barrings. Not dun 1 causes primitive markings without the dun dilution, and not dun 2 causes no primitive markings or dilution. A bay horse with dun is called a bay dun. A chestnut horse with dun is a chestnut dun or a red dun. And a black horse with dun is called a black dun or grullo. A horse with ND1 is referred to by its colour, then with ND1. So chestnut with ND1, bay with ND1, and black with ND1. And dun is found in many horses such as quarter horses, stock horses, and Icelandic horses. Champagne is our next dilution gene, and champagne dilutes red pigment to a goldish colour and black pigment to a brown colour. Champagne can also cause the skin to dilute to a pinkish colour. It doesn't matter if there's 1 copy or 2 copies of champagne, the result is basically the same. So a chestnut horse with champagne is a gold champagne, a bay horse with champagne is an amber champagne, and a black horse with champagne is a classic champagne. And champagne is found in many breeds, including quarter horses and mustangs. The next dilution gene is silver, and silver dilutes black pigment only. Silver gene is associated with a disease called multiple congenital ocular abnormality syndrome, which is more severe when they have 2 copies of silver. So usually we just call it the colour plus silver. So a chestnut with silver will look exactly the same as a chestnut without silver, and it's just referred to as chestnut with silver. A bay horse with silver can be called a silver bay and has the light mane and tail. And a black horse with silver is called a silver black and has the white mane and tail but maintains the black body. Silver is found in many breeds such as Icelandic horses and Rocky Mountain horses. The final dilution gene is mushroom. Mushroom is a gene that dilutes red pigment to a sepia colour. So originally it was thought that only chestnut horses with 2 copies of mushroom would show the mushroom colour. However, recent testing has shown that bay horses may also exhibit mushroom colour on their bodies if they have 2 copies of mushroom. Mushroom is found exclusively in Shetland ponies and miniature horses with Shetland pony ancestry. Next we have the white patterns, and there's quite a few of them. The first white pattern is Tobiano, which is a white pattern that causes white all over the horse's shoulders and hips and can cross over the horse's top line. The head and barrel of the horse are often coloured and the legs are often white. The tail can also be partially white. Tobiano is noted as capital T/O/N and is found in many breeds. The next one we have is frame, which causes white markings along the body of the horse, leaving a frame of colour around the edges. The white is often jagged at the edges and can cross over to the other side under the barrel. 2 copies of frame result in incomplete digestive tract, and the foals don't usually survive more than a couple of days post-birth. Frame is found in quarter horses, paints, miniature horses, and thoroughbreds. Sabino is the next white pattern. It causes white markings on the legs and white ticking along the underside of the body, neck, head, and face. It can range from very minimal to extreme expression, with horses with 2 copies being over 90% white sometimes. Sabino is noted as SB1/N and is found in many breeds. The next white pattern we have is splash, which causes white markings up the face and belly. and as though the horse is splashed through white paint. There are multiple variations of the splash gene, known as SW1 through to SW8, and the amount of white can vary from only a small amount on the lower legs to almost everywhere except a small portion of the back, depending on the combination of genes involved. Splash is found in many breeds, with different variations being specific to some breeds. Dominant white is a group of white patterns that are all variations of the KIT gene. They vary greatly in the amount of white they produce, from white face and leg markings to covering the entire horse. Many of the dominant white mutations are breed specific. Like in the picture here, we have the white thoroughbred, which is either white 5 or white 22. But other breeds with dominant white are quarter horse paint, Appaloosa, Pony of America, Shetland miniature, and pretty much every other breed. Some variations of dominant white are expected to be lethal as an embryo if they get 2 copies, because we've not actually found any to be genetically tested to have 2 copies of that same dominant white. Roan is a white pattern that results with white hairs mixed evenly throughout the coat, everywhere except the face and lower legs. Roan horses can appear darker or lighter depending on the season, however they are always born with the roaning. One obvious sign of roaning is the inverted V on the front of the horse's legs, where the roaning joins a non-roaning portion of the limb. The exact gene for roan hasn't yet been identified, though marker genes have been identified in quarter horses and paints. Now these 2 are Appaloosa specific white patterns. So leopard is a white pattern that causes the coat to be mostly white with coloured spots throughout. It causes mottled skin, striped hooves, and white sclera. The amount of white can increase as the horse ages. 2 copies of the leopard can cause congenital stationary night blindness. And as I said, leopard is found in Appaloosas and Appaloosa-derived breeds and is sometimes called Appaloosa spotting. Pattern is a modifier of leopard and controls the amount of white present on the horse's coat. 2 copies of pattern boost white more than 1 copy. And pattern causes varnishing, which progresses as the horse ages. Pattern is highly variable in its expression and often changes with seasonal shedding. Lastly is grey. It's not a dilution gene or a white pattern. However, it does modify the horse's entire coat colour and result in a nearly white horse. The grey gene causes progressive colour loss by excessive pigment production, then depigmentation until the horse appears nearly completely white. Grey horses have a high instance of melanoma when compared to non-grey horses, and grey is found in pretty much every breed. So now we know what the genes resulting in colour are, we can use the genes of the parent to work out what the potential offspring could be. So if we know the genetics of the parent, we can work out the potential genetics of the offspring using a Punnett square. Using grey as an example, if each parent has 1 copy of grey, you can use the Punnett square on the right to work out the potential offspring. So we can see we've got 1 parent on the left and 1 parent along the top, and the potential offspring in the centre light green boxes. From our Punnett square, we can work out that the genotype, there is a 25% chance of 2 copies of grey, 50% chance of 1 copy of grey, and 25% chance of no copies of grey. Now for the phenotype, we can see that there's a 75% chance that the foal will be grey, and a 25% chance that the foal will be not grey. This is a fairly straightforward Punnett square with only 1 gene. When you start getting multiple genes, things like buckskins or say you've got a bay going grey, where you've got 3 or 4 different genes on each parent's side, can get quite complex and you might have 16 columns going across, but the actual process is still the same. So here's the list of references I used. You can use the name and years to match each reference to the ones on the slides to see where I got the information from. And for the images, thank you to the members of the Facebook group Equine Coat Colour Genetics who provided many of the images along with the verified genetic test results to ensure that they portrayed the actual colour they said they were. And that's the rest of the references.